Hi, and welcome to your background video for the Milk and Interesting Mixture Lab. Before attempting this week's experiment, you should be able to describe the 4 components of milk that we will be separating and work towards understanding how the principles of solubility and polarity allow us to do this. The 4 components of milk that we will be looking at today are protein, fat, carbohydrates, and phosphates. Before we get too far into the actual background, let's start with some definitions. First, a simplified explanation is solubility. Solubility is the property of a solid, liquid, or gaseous solute to dissolve in a solid, liquid, or gaseous solvent to form a solution. If a solute disappears into a solvent, just meaning that you can't see the solute particles anymore, we say that the solute is soluble in the solvent. If you can see solid after sufficient stirring and heating, we say that the solute is insoluble in the solvent. Second definition, polarity. The polarity of a substance describes whether or not a molecule or substance has a positive area, a negative area, or both. If the substance has both, we say it is polar. Take water, for instance. Because of the shape of the water molecule, there is an obvious positive area and an obvious negative area. If a substance does not have both a positive and negative area, we say the substance is nonpolar. Look at our example, methane. Methane contains only carbon and hydrogen, which have about the same polarity, so the molecule doesn't have 2 different polar areas. Methane is nonpolar. All right, with those definitions out of the way, we can move on to the background for the activity for this week. The first of our 4 components we mentioned was a protein. A protein is a large organic polymer made up of amino acids which are linked by peptide bonds. Amino acids are a large group of molecules that share the same generalized structure. The presence of an amine group, a carboxylic acid group, and a functional group, which we represent by the R that is unique to each amino acid. These functional groups differ in polarity, which allows proteins to have some pretty unique solubility features. The peptide bond forms between the carboxylic acid of one amino acid and the amine group of another. When placed in water, proteins are able to fold up in a way that allows them to become soluble. This tertiary structure results from the nonpolar or hydrophobic functional groups being surrounded by the polar or hydrophilic functional groups, which are the ones that actually come in contact with the water. Proteins can be denatured, which means that the tertiary structure of the protein is disturbed by heat or chemicals like acids, which cause it to unfold. So now the hydrophobic groups can come in contact with the water, which lowers its solubility and results in the protein precipitating from the solution. A fat is a lipid composed of a glycerol and long fatty acid side chains represented here by R. Just like with the amino acid, the side chains vary and are what identifies each type of fat. However, even though those side chains can vary, they're composed mostly of carbon and hydrogen, so we consider fats to be generally nonpolar substances. You may have heard of saturated and unsaturated fats. This refers to the number of double bonds present in the side chains. Unsaturated fats have more double bonds between the carbon atoms, and saturated fats have mostly single bonds. The double bonds change the shape of the fat so they can't stack up on each other. This stacking is what makes saturated fats so bad for you. These large hydrocarbon side chains help make the fat molecule hydrophobic, which means that they are not very soluble in a polar solvent like water, but that they are soluble in a less polar solvent like acetone. Sugars are a type of carbohydrate that have the formula CnH2OM. A monosaccharide is the basic unit of a carbohydrate. Fructose, glucose, and galactose are all monosaccharides. Disaccharides are a type of sugar made by joining 2 of these monosaccharide units together. Sucrose, maltose, and lactose are all disaccharides. Lactose is the main sugar found in milk and is made of 1 glucose and 1 galactose component. If you look closely at this molecule, you will see that there are many hydroxyl groups on the molecule. These groups are hydrophilic, which makes lactose very soluble in water. Lactose is an aldehydic sugar, which allows us to use the Benedict's test to confirm the presence of the sugar. If the sugar is a ketone sugar, like fructose, this test will not work. Phosphate is the last component we will separate. Unlike the previous 3 components, phosphate is not a source of energy. It makes up part of your bones and can be used as a buffering agent in your cells. You will be using a calcium solution to precipitate the phosphate out of solution. The phosphate you will collect will actually be in the form of calcium phosphate. Next week, you're going to weigh this calcium phosphate, and you'll have to work from grams of calcium phosphate to moles of calcium phosphate, from that to moles of phosphate, and eventually to grams of phosphate. After watching this video and reviewing your lab manual, you should be ready to watch the technique video and explain how each exercise works and allows us to separate the components.